There we go. Okay, happy Friday. Let's see, a couple announcements. Number one, I noticed, or I forgot to mention that if you've been looking at your homework assignments, there are some homework questions with chapter 8, which is just a review chapter. So review of macromolecules, lipids, some biochemistry in there. I assigned those questions just as review. So if you feel maybe a little bit weak in those areas and you want to look through it, but by all means, it's a resource there for you to look at. Yeah.
Do you have an idea when the Unit 1 homework answers are going to go?
So you should be able to go into Gradebook and see how you did on the homework that was due at the first exam. I have trouble knowing exactly what you can see. So if you have trouble finding that, then come and sit down and we'll take a look at that. But I believe that I have graded everything now, so it should be that number that's in there is what you got. So barring any adjustments. So yeah, take a look at that sometime. And again, the chapter 8 is just, optional. You can be working on the kind of a shorter unit, the eukaryotic with the yeast and the fungi and the malaria prokaryotes, as well as chapter 7 on growth. There was a few of you that forgot to do the little part of chapter 7 that talked about the growth media. So just be aware of that. I tried to remind you, but the rest of chapter 7, and hopefully we'll come pretty close to finishing chapter 9, metabolism today. So got some pathways to go through today. Also want to just remind you, hey, I was on the way in to work yesterday morning, they were talking about the flu, and usually an indicator of how bad a flu season is going to be if we look at how was the flu season in the Southern Hemisphere, because they're just coming off of winter. It was really bad. So that's a good indicator that there's going to be a lot more flu cases this year. Hey, this would be a good weekend, maybe go get your flu shot. You can walk in a Walgreens and I believe it's no cost. They'll give it to you. So just wanted to throw out that public service announcement as well. And also, before I get to a review question, hopefully maybe you've been thinking about that already. I just wanted to revisit, we had a very good question last time. We finished talking about the pentose phosphate pathway. We talked about how this is a pathway that a cell would use, both a eukaryotic cell and a prokaryotic cell, if it needed precursor molecules for to make some other compound for biosynthesis, and how this is a way for them to get 5-carbon pentose sugars for nucleic acids, as well as this 4-carbon erythrose 4-phosphate, which is used to make aromatic, the ringed amino acids. Bacteria do that. Humans do that as well. I wasn't sure about that last time. There are what's called essential amino acids that we have to get from our diet that we can't make, but there are some that we can synthesize as well. And there are some aromatic amino acids that we are able to synthesize, again, with this precursor. So just to clarify that. And I realized that I had a form of this question that I tweaked a little bit up before. So that's why I don't know if you noticed that I changed it, actually changed one of the answers. But Entner-Doudoroff, so we talked about that last time. Okay, which of the following, which of these is true about Entner-Doudoroff? Do you need a second to think about it, or have you been thinking about it already? Okay, I'll give you like 20 seconds to read it again. All right, so let's see what we can cross off. Okay, A, it is not the same pathway. And Embden-Meyerhoff-Parnas, that's what's typically just referred to as glycolysis. But since we talked about other glycolytic pathways, entner is considered, we get sugar splitting as well, but it's not the same thing. Is it common to most forms of life? No, we said this is only some bacteria can do this. It's usually because they're missing an enzyme of Embden-Meyerhof harness pathway. So that takes care of this. It's not carried out at the same time. Organisms are either doing one or the other. It's not an anabolic pathway. It's catabolic. You're starting with glucose and you're ending up with smaller, with 3 carbon pyruvates. So that's breaking molecules down. So the true one there, it does produce half the ATP that we get from Embden-Meyerhof harness glycolysis. All right, so little review there. Let's move on in our look through respiration. Okay, so thinking back here, assuming there's some final electron acceptor available, it will continue those 2 pyruvates that we get from some either entner or Embden-Meyerhof harness pathways. What happens to those pyruvic acids? Well, it turns out that for that pyruvic acid to enter into the Krebs cycle, the Krebs cycle only accepts 2 carbon compounds. So there has to be a little transition step there. A carbon is lost, call that a decarboxylation when it's lost just to carbon dioxide. And that gives us a 2 carbon acetate. In order for that acetate to be, again, attached or flow into the Krebs cycle, a coenzyme facilitates that. So this coenzyme attaches to acetate. In that process, we get some electrons there. We get a little reducing power, which will get changed into ATP at the electron transport system. But then we end up with acetyl coenzyme A. So that's a 2 carbon compound, then that can be shunted into the Krebs cycle. Again, really simplified view of the Krebs cycle here. We have 3 different names for this. The Krebs cycle, which gives homage to the person that elucidated it, Hans Krebs, back in the 1800s, I think. The citric acid cycle, which they've labeled it here because the first compound that's made is citric acid. That is shown on the next diagram that I'll show you. And also the tricarboxylic acid cycle, or TCA. Okay, those are all referred to the same process. Sometimes we call it tricarboxylic acid cycle because some of the intermediates have 3 carboxylic acid groups. That's COOH. Okay, so if we looked at structures, that would be a little bit more obvious. Think about where this is taking place. For prokaryotes, it's going to be in the cytoplasm. This is a little bit thick. Same as glycolytic pathways. And eukaryotes, it's going to be in the matrix of the mitochondria. So we didn't really talk about the structure of the mitochondria, but of course there's a plasma membrane around the mitochondria. And then there's these inner folds called cristae. And then, so the matrix is the very inside there of the mitochondria. Okay. All right. So, like I said, this is a simplification. What are we getting out of the Krebs cycle? We're getting lots and lots of electrons. So NADs are being reduced to NADHs. Remember, they're carrying a hydride ion. They're actually carrying 2 electrons and a proton. We get 1 FAD carrying 2 electrons and 2 protons. We're losing 2 carbon dioxides. This is per acetyl-CoA. So if we started, remember we started with that 3-carbon pyruvic acid. We lost a carbon in that transition step, and then we lost 2 more here in the Krebs cycle. So all of those carbons that we started out with, the 6-carbon glucose, now have been lost. Kind of interesting. Some organisms can't afford to lose so much carbon. They need that carbon to build structures. So there are modified citric acid cycles where organisms kind of just have some modifications so they don't lose all that carbon. The counting here gets a little bit tricky. You have to pay attention to, are we counting per, like here, they're counting per acetyl-CoA. Oftentimes, we might count per pyruvic acid, and then we have to include this NADH here. Sometimes we count per glucose, and knowing there's 2 pyruvic acids per each glucose, so you have to double it. So when you're counting up what you get out of it, you have to pay attention to what you're including in that count. But let's say net gains per acetyl-CoA, kind of like they count it here. 3 NADHs, 1 FADH2, and 1 ATP. It says ATP or GTP, pretty much the same thing. GTP is just a little different energy currency of the cell. And that's by substrate level phosphorylation. It's not as obvious that phosphate comes from the substrates here if we were looking at structures, but we do consider it substrate level phosphorylation. Okay. Also, we could write in here 2 carbon dioxide. So, all right. So importantly, these electrons. Remember, we said last time it's all about the electrons. Those are going to get converted into lots of ATP at the electron transport chain. I did want to show you, they added a few details in their second figure in OpenStax. And like I've emphasized before, don't memorize those intermediates. But here we start with the 2-carbon acetyl-CoA that attaches to the 4-carbon oxaloacetic acid to make citric acid or citrate. Okay, that's where the name of one of the names comes from. And this notes where there's some kind of branch points if we were interested in looking at synthesis reactions. So we like to think about these reactions kind of always following the arrows, but they're actually in the cell kind of all over the place. Like it depends on what the cell needs. Oh, now it needs this. So this 6-carbon citrate is going to branch off to make some fatty acids. We're not going to really delve into that and worry about that. I just wanted you to kind of appreciate that there's lots of different branch points there that we could follow. Right, let's see where the NADHs are formed here, and up here. Here's where we get the electrons that are carried by FAD. Okay, so that's pretty much, again, lots of organisms doing Krebs cycle. It's part of any kind of respiratory pathway. Okay, so back to the overview, we've come through some glycolytic pathway and looked at the Krebs cycle. Now we're ready to take a look at what happens to these electrons that come off of glycolysis and the Krebs cycle. Where do they go? They're going to go to some electron transport chain, or your book usually calls it electron transport system, ETS. This is just a series of redox reactions that are always going to take place in the cell membrane. And it's very important that there is, in fact, an intact membrane. And hopefully I'll point out why that's the case in a minute. All right, so a couple different ways that I want to show this. This graphic is good at pointing out some things, and the next graphic is a little bit better at pointing out some other things. So first note, this would be the outside of the cell, and this would be the inside of the cell, assuming we're talking about a prokaryotic cell. So again, prokaryotic cell, this is taking place in the plasma membrane. And it's really important that that is in the membrane because there has to be like an outside and an inside. Eukaryotes, this is going to be in the inner membrane, so that mitochondrial matrix membrane. All right, so what is this? This is a series of redox reactions. Okay, whenever you have a reduction, you have to have an oxidation. They always go right together. Where you have some final electron acceptor. So the reason that this is pictured slanting down, that's to show the energetically favorable way for the electrons to get past. Okay, so if this up here is the top of the hill, okay, and this is the bottom of the hill down at the bottom, that's the way the electrons are going to roll. We can put electrodes in a cell and measure that. So we know that this molecule, NADH, very easily gives up its electrons. And whatever the final electron acceptor is, really wants to grab electrons from other molecules. Okay, so that makes it energetically favorable. I'm going to show you a redox tower in a little bit that will hopefully explain that a little bit. But to start out with here, NADH is oxidized back to NAD. Okay, so that's really important. Now the NAD carrier can go back and get more electrons from those pathways. We'll keep those pathways going. Okay, if there's not NAD to go back and take off electrons off those pathways, then those oxidation reactions will stop. Okay, so that keeps it going. And this is where we get the name oxidative phosphorylation, because NADH is oxidized. Okay, those electrons and protons, what happens to those that are coming off the NAD? The electrons get passed through a series of electron carriers. Okay, so I listed some of them down here, electron carriers. And don't memorize the order of these, because that can be different from organism to organism, but you should kind of recognize, oh, a flavoprotein. Oh, that's an electron carrier, an electron transport system. Cytochromes. Okay, those are electron carriers. Quinones, iron sulfur proteins. Okay, the one that maybe you really should recognize, and there's at least one homework question about this, the very last one here, when the final electron acceptor is oxygen, then the final carrier here is called cytochrome oxidase. Okay, that's the one that passes its electrons off to oxygen. Okay, so it's kind of a famous one, cytochrome oxidase. Sometimes organisms will have a little different cytochromes. For example, there's a test that we do to identify bacteria that tests, oh, do they have this particular cytochrome? There's a huge group of gram negatives that don't have it, and there's a little smaller group of gram negative organisms that do. We call that the oxidase test. But again, that can be variable. Kind of recognize what an electron carrier is. Most of them are passing both electrons and protons. What do they do with the protons? You kind of see this here. The protons are being pumped outside of the cell. Okay. And that starts forming a proton gradient. So if you think back to the picture that I showed last time of the dam holding back the lake. Okay, so the lake is lots of potential energy when it flows through the turbines of the dam. It's creating electricity. Okay, this proton gradient that's building up here, that's like the lake, because it wants to diffuse from places of high concentration to low concentration. So there's one channel in the membrane where those can diffuse back into the cell, and that powers the phosphorylation of ADP to ATP. Okay, so that one channel, it's not shown here, it's shown on the next one. But this is a little bit misleading when you see these yellow ATPs here, because that's not really the spot where ATP is generated. That's just about the spot where enough electrons have been passed and protons pumped out to generate an ATP. So that's a little bit misleading. FAD comes in the pathway down a little further down than what NADH drops its electrons off. So we get a little less ATP per each of those electrons that FAD carries. Okay, so that kind of plays into the equation later on. Let's see, okay, all right, so the next slide, again, it shows, I want to point out a few more details. Here's this cytochrome oxidase. So again, assuming the final electron acceptor is oxygen for now, that's cytochrome oxidase, where we've got actually oxygen has to get split to make water. And that doesn't always happen just right. Sometimes mistakes are made, electrons are not distributed evenly, and maybe up to about 2% of the time, we get something called superoxide instead of water. Again, because those electrons didn't get distributed equally or correctly when oxygen is split. We talked about superoxide on Monday. It's one of those reactive oxygen species. The cells have to have a way to detoxify. Otherwise, that superoxide is, if we said it was an oxidation agent, it's grabbing electrons off of other molecules and causing a lot of cellular damage. So cells have to have an enzyme to detoxify that, like superoxide dismutase. They end up with hydrogen peroxide that also has to be detoxified, but that's where some of that toxic oxygen products come from. In addition, sometimes some electrons will leak a little bit back inside the cell rather than getting passed correctly, so that they can form superoxide just from those kind of leaking electrons. Okay, so that proton gradient that had built up, sometimes we refer to that as what's the word that I'm looking for? Proton motive force. Okay, so that's a term that just makes us think about how there's a huge gradient. That's a lot of potential energy outside of the cell, and where they are allowed to diffuse back in, again, this is down their concentration gradient, not spending energy to bring protons into the cell, is this enzyme called ATP synthase. Okay, so it's really important to have an intact membrane, otherwise those protons can just diffuse in anywhere. We only want them to come through that one spot And then that powers the phosphorylation of the ADP to ATP. Okay. That process is also referred to as Chemio Osmosis. I think there's only one O, osmosis. Sorry, my handwriting's really messy there. Chemio Osmosis. So those two terms, proton motive force and chemio osmosis, they're really kind of closely related. Question. Is that the same as facultative diffusion? Facilitated diffusion. Which that does not require ATP, if I remember correctly. So yeah, I guess it would be. Yep. Yep. Good point. So right, the diffusion of the ions, it's facilitated by a particular enzyme. And again, that process of those ions diffusing in, we also refer to that as chemio osmosis. The next slide just kind of brings up those two terms and just parses them out a little bit better. That proton motive force, that's the buildup of protons outside of the cell. Again, that process where they're basically, you could think about diffusing or osmosing back in. So the process by which ATP is generated because of that proton motive force, we call that chemio osmosis. All right, so here's your book's kind of sum up of respiration. Again, I don't get too caught up into memorizing counts of this or that. And it's kind of interesting the way they present this here. If you think about the net ATP molecules made during, we'll call it they call it by oxidative phosphorylation. So that's at the electron transport system. So they, like, for instance, in the Krebs cycle here, we get 6 NADHs. So that converts into 18 ATP. So they're assuming that for about every 3 NADHs convert into 1 ATP, and about every 2 FADH2s will convert into 1 ATP. So that's where we get from 6 NADHs to 18. 6 times 3 is 18. And then here for every FADH, we get 2 ATP. So that makes 4. Okay, so that's the way they count that. And then in this, and just, just reminds us, well, for glycolysis, we had to put 2 in and we got 4 out. So that's a net of 2. We did talk about that. The theoretical maximum of ATP, keep in mind that's very theoretical, because especially in prokaryotes, they use that proton motive force to power things like flagella movements. So they don't actually use ATP to move their flagella. They use that proton motive force. In eukaryotes, they use ATP. It can be very different. Prokaryotes also can use proton motive force to power things like transporting molecules into the cell. So they probably never reach that theoretical maximum. And in addition, this is considered a little bit different for eukaryotes because they have to spend a little bit of ATP to transport NADH from glycolysis into the mitochondria. So in eukaryotes, theoretically, it's slightly different. Okay, so now I want to think about what if organisms are using some other electron acceptor other than oxygen. Okay, so that's something that you and we never do that. It's true that we can do some fermentation. That's a separate process that we're going to get to in a minute, and hopefully you understand the difference between anaerobic respiration and fermentation. Those are way different processes. Okay, so this is only something that bacteria do, not us, because they have certain enzymes that make it able for them to do that. Okay, so if there's an electron acceptor, something other than oxygen, it's usually going to be some inorganic ion. Okay, so remember, something's inorganic if it doesn't have carbon-hydrogen bonds. Okay, and this is usually something that comes from outside of the cell. Okay, something from the environment. Okay, so what's some examples of electron acceptors other than oxygen? One is nitrate. Okay, and I mentioned that one first, and we're going to look a little bit more closely at that one because the enzyme that reduces nitrates is one that you're going to test for in lab. So 2 electrons plus 2 protons. The enzyme there is called nitrate reductase. And what you end up with is nitrite and a little water. Okay, so that's a reduction reaction. Sometimes it's hard to figure out how is that a reduction reaction. You have to go in and kind of look at oxidation states to understand that. Usually when you lose an oxygen, usually that's a reduction. But let me show you, I'll come back to this page in a second. But here's an example of hopefully to illustrate how organisms can use something like nitrate. Or here, the enzyme nitrate reductase is that big green blob there. Okay, it's an enzyme that's right there, right in the electron transport system of some bacteria that allows them, again, to use nitrate to accept those electrons. Okay, so that's an enzyme we can't make. The Enterobacteriaceae, that's a huge family of bacteria. Sometimes we refer to them as enterics. E. coli is an enteric. Okay, so it's just an example of an organism that can use nitrate. Another example, sulfate. Okay, an inorganic ion usually end up with hydrogen sulfide when organisms can do that. That's the gas that, if you've ever been to Yellowstone, you smell that like rotten egg smell, that's hydrogen sulfide. That's a microbial process, and that's another end product that eventually in lab you'll be testing for to see if organisms can respire anaerobically. Carbonate, another inorganic ion, I guess -2 there. Organisms that can use carbonate usually will end up with methane. Okay, it's some archaea that can do that. We end up with methane, archaea called methanogens. So here's an example. You think about, well, where do we find bacteria that are respiring anaerobically? Okay, places where there's no oxygen, so swampy bogs, landfills. If you've ever been to a landfill, they are, continuously covering up trash with dirt and burying it. Deep down in there, there's very little, no oxygen. You have to vent that. Otherwise, methanogens that are making methane, it becomes an explosion risk. You have to vent that, and most of the time they just burn off that methane that's produced, but occasionally they will collect it, and then you can use that for electricity. So it's not done on a wide scale because it's kind of expensive to produce electricity that way. But I always like to show examples where we're using bacteria to help us in some way. Okay. All right. So back to the overview, just remember, again, anaerobic respiration, they're doing glycolysis, Krebs cycle, and an electron transport chain. It's just they're not using oxygen. They're using something else down here, like maybe nitrate, for example, or sulfates. One other point I want to make about anaerobic respiration, and that all involves kind of a basic understanding of something called a redox tower. And we're not going to do any calculations with this. I'm going to leave that to my biochemist colleagues. But there's one major point I want to make, and then the redox tower kind of hopefully explains why that's true. But organisms that do anaerobic respiration, they are always going to get less oxygen from that, sorry, less ATP from that than if they had done aerobic respiration. Okay, so less ATP always produced from anaerobic respiration. Compared to aerobic respiration. Okay, the reason why. I mentioned before, we can put electrodes in cells and measure voltages. What we can measure here is something called E naught prime. So E with a little dash and a little zero there, that's pronounced E naught prime. We can actually measure the tendency of molecules to give up electrons. And if that tendency is very negative, that means that they really want to give up their electrons. They easily let them go. If that E naught prime is positive, that means a molecule really wants to get electrons from other molecules. So if you look up here, NADH, okay, that has a very negative E naught prime. It gives up those electrons pretty easily. That's why it's kind of at the beginning there of the electron transport chain. And then oxygen really wants to get electrons. So it's really positive. So if you measure the distance here between the molecule that gives up the electrons and the molecule that gains it, it's a really long distance. We get a lot of energy per electron when aerobic respiration is done. So that's how the greatest change in the reduction potential produces the most energy. And we could calculate that. That's kind of what these numbers are here. We could calculate that. If anything other than oxygen is used, so say like here's sulfates, or say nitrate is used, it's just going to be a shorter distance. Okay, so you're not going to get quite as much ATP. Incidentally, this is how we know the order of the electron carriers here, the cytochromes, ubiquinone example, for example, because we can measure their tendency to give up electrons. Okay, so always going to get less AT from anaerobic respiration, but these organisms, these bacteria, can live in places where there's no oxygen. Okay, so that again gives them the advantage. Maybe they can live in places where there won't be as much competition, perhaps. Okay, so what kind of organisms will do anaerobic respiration? We talked about oxygen requirements earlier in the week. Facultative anaerobes can either do aerobic respiration or some kind of metabolism where oxygen's not necessary. It could be anaerobic respiration. It could be fermentation. Aerotolerant anaerobes, strict anaerobes are probably respiring anaerobically, maybe doing fermentation as well. So what's the difference between anaerobic respiration and fermentation? The difference is the fate of pyruvates. Okay, that molecule that we ended up with at the end of glycolysis, what happens to pyruvates? Okay, back to that overview. Here's our pyruvic acid right here. If there is no suitable electron acceptor down here, then it's not going to pass through these pathways. Okay, so if there's no electron acceptor down here, the cell has no choice but to go into some fermentative pathway. Okay, so glycolysis is considered the first step in both respiration and fermentation. And so what's accepting electrons in a fermentative pathway? It's usually going to be some organic molecule. So in fermentation, there's an organic electron acceptor. It's usually something that's already in the cell. Okay, quite often it's going to be pyruvate. And the main points of this fermentation, any fermentative pathway, so let's say the point of fermentation is to oxidize NADH back to NAD. Okay, these NADHs that are coming off of glycolysis, they need to dump their electrons off so that the NADs are available to take electrons off of glycolysis. Okay, if these NADHs can't cycle back to NAD, then glycolysis will stop, because you can't have the oxidations of glycolysis without something else getting reduced. Okay, so that NADH has to cycle back. There's often no ATP produced by any fermentation pathway. That's not always the case. Sometimes you get a little additional ATP, but at least the organism still gets the ATP from glycolysis. Okay, so at least it keeps glycolysis going because it's cycling back the NAD. All right, so let's look at a simple fermentation pathway. And I'm not crazy about this figure because it makes it look like, well, the pathway could go this way or it goes both ways. This is showing several different fermentative pathways, and very often an organism only has one pathway that they'll use. It depends on the enzymes that they have. But the very simplest one is this lactic acid fermentation. That's the one that we can do in our muscle cells. Okay, you know, you're running a 400-meter race, you know, you get to the 300 mark, that lactic acid is building up in your muscles and it gets hard to move. Okay, so you know, we can do that a little bit. There are some bacteria that all they do are lactic acid fermentations. There may be oxygen present, they ignore it because all they do is fermentation. All they do is the simplest kind of fermentation. So basically, pyruvic acid acts as an electron acceptor. Here's it accepting electrons. And then what we end up with is lactic acid. So you can see that it's very similar to pyruvic acid. Okay. And you know, no gas produced, anything like that, just get lactic acid. So this is for bacteria, they are almost always going to have acidic end products. And that's often how we test to see if fermentation is taking place. We look for a pH change. Okay, acidic, lactic acid sounds acidic, right? Some organisms, like yeast, for example, though, will do an alcohol fermentation. Okay, we talked about that. How yeast can, give us useful things like beer or wine, for example. Bacteria usually don't do that. Again, this is the alcoholic fermentations are usually just yeast. Okay. There are many other possible fermentation pathways. And I didn't post this because I didn't want anybody to get worried that they had to know it. But I always like to show and see how bacteria are useful to us. We can figure out what fermentation pathway they're doing, and that helps us identify them. We're going to do that later on in the semester in lab to test to see if organism does what we call a mixed acid fermentation, where they end up with acetate as one of the end products, or 2,3-butane-diol fermentation. There are different organisms that can do that one. There are organisms that, like propionibacteria, that end up with propionate when they do their fermentation pathway. Okay, those are the organisms that make Swiss cheese for us. That's a distinctive flavor in Swiss cheese. This carbon dioxide that comes off, that's the holes in Swiss cheese. We often see gaseous side products of fermentation pathways. If you were in lab, maybe your lab professor talked about C. diff when you were talking about spore formation. Clostridium, a lot of the Clostridia here will do a fermentation where they get butanol as their end product. That is the smell from C. diff diarrhea. That's supposed to be a very distinctive smell. People say the whiff of the diff, like they can identify whether patients have C. diff diarrhea based on how it smells. It's supposed to be a very distinctive smell. So we can spend a lot of time on bacterial fermentations. I had a student say to me once, I just don't think microbiology is all that interesting. I said, look at this. This is what the whole deodorant industry is based on, bacterial fermentations. Warm, moist places in your body, lots of bacteria making really stinky end products. So that is fermentation. Again, this doesn't match up very well next week in lab. Yeah, next week you're going to be doing these fermentation tubes, where basically you start with these purple broths. There's a pH indicator in there and a sugar, and we can tell if our organism is able to metabolize, able to ferment that sugar, because if we see a big pH change, it'll change to yellow, and then we can say, oh yeah, it's fermenting that sugar. So you're going to do that test next week. And I just wanted to point out that, again, because of bacterial fermentations, we get lots and lots of useful products. So this is not to memorize, just to say, hey, this is interesting. Bacteria are useful in your life. You get things like Swiss cheese, soy sauce, yogurt, alcoholic beverages, even things you don't think about, like nail polish remover, right? Acetone, isopropanol, those come about as a result of microbial fermentations. Of course, vinegar is acetic acid. So lots of useful products due to fermentations. Okay, we're going to wrap up. There is this little, every once in a while I post a little chart. This is maybe useful to kind of cover up and test yourself. Remember, be doing that as much as you can. Review. We'll finish up metabolism, thinking about what if proteins or lipids are metabolized, just like 5 minutes, and then we'll start genetics on Monday. So do a little homework this weekend and do something fun to have a good weekend.